I'm just going to start with a short story. So in 1958, the chief surgeon at Denver General Hospital, a man called Mr. Ben Eisman, was faced with a situation that he'd sadly seen many times before. He was placed in charge of the care of 4 patients who had developed heavy diarrhea, stomach cramps, and high fever after having been given antibiotics post-surgery as a means of prophylaxis. Now, the other doctors in the hospital had exhausted all known treatment options, and Mr. Eisman, as the chief surgeon, had been placed in charge of their care to make a decision about whether or not to put them on the palliative care pathway, in essence, to try and make them as comfortable as possible before they passed away. I think I perhaps know what to do here. Or maybe not. Anyways, so Mr. Eisman, not really sure what to do, had a bit of an epiphany. And he was a bit of a pioneer, much like Mr. James Kinross. And he decided that he was going to try and transfer fecal material from a donor into their intestinal tract. And remember, these were also the days where if a doctor wanted to do something, they just kind of did it. Obviously, it's much more rigorous right now. Now, remarkably, all 4 of those patients were cured of their infection and walked out of the hospital shortly after. And that was the first instance in modern medicine of this treatment, which we now call fecal microbiota transplantation, being used in the hospital setting. And he published his results in the journal Surgery in 1958. And I don't have the slide yet, but I think he said something along the lines of, this rational therapeutic deserves further intervention or further exploration or further research. Now, these days, FMT, as it's now known, involves the same kind of thing. We transfer fecal material, stool, poo, whatever you want to call it, from donors into the intestinal tract of patients suffering from diseases and infections associated with imbalances of bacteria. Now, the paper that Mr. Eisman published had a title saying pseudomembranous colitis, which is a clinical manifestation of C. difficile. So it's highly likely that those patients were treated for C. difficile, and today that's the primary indication, if you like. FMT can be administered through a tube that goes up the nose and into the intestine. Sounds a bit yucky, and you'll hear just in a second. The first randomized controlled trial that was published used the same technique. Or it can be done through a tube that goes up the backside. And there we go. So, right. Oh, I can see it, which is good. So you're imminently about to see it. The slide, which is going to come up very soon, presents the first randomized controlled clinical trial of fecal transplantation in the modern era in a disease indication. And the title of this paper is Duodenal Infusion of Donor Feces for Recurrent C. difficile. Now that clearly is a title we're talking about. And back then, it really made the news. Now I remember reading that paper and going, oh my God, they did what? Because essentially that means infusion of fecal material up the nose and into the first part of the small intestine to treat a disease which is historically very difficult to treat, especially when it's in its recurrent form. But what was most remarkable about that paper was the data that they presented. When they compared fecal transplantation to then the standard of care, which is an antibiotic called vancomycin, over 90% of the patients who received 2 fecal transplants were cured clinically and left the hospital within 48 hours. The trial, sorry, I didn't realize it wasn't up. Beg your pardon. So this is the trial here. And in essence, actually, this trial was stopped early because of an interim futility analysis. And it was deemed not to be ethical to continue giving the patients with C. difficile infection vancomycin because this other intervention, fecal transplantation, was so much more effective. And following on from the pioneering work of these Dutch researchers, there's been a substantial increase in both the quantity and quality of evidence available supporting the use of FMT in recurrent C. difficile infection. To give you a more broad overview, about 15,000 people every year get a C. difficile infection, and around about a quarter of them will experience recurrences. Once you've had 1 recurrence, your risk of having another recurrence goes up dramatically. Once you've had 2, your risk of having a third or fourth goes up to over 60%. Now, there's been 10 or over 10 randomized controlled clinical trials now of FMT in recurrent C. difficile infection, reporting broadly similar results to Van Nood et al. When they published their paper in the New England Journal of Medicine. And in light of that, fecal transplantation now features on most of the clinical care guidelines for patients with C. difficile infection around the world. In the UK, it was endorsed by the National Institute for Health and Care Excellence, NICE, in 2014. Shortly after that, Public Health England started endorsing it. Then Scotland was to follow, and this is just a little snippet from an American society to show you that transatlantically we're all thinking the same thing. Now, one criticism up until relatively recently was that there hasn't been a clinical trial which has compared fecal transplantation to newer antibiotics, which have a narrower spectrum, if you like, of activity. Vancomycin pretty much obliterates all gram-positive bacteria and is very non-specific. Now, it does have excellent specificity and potency against Clostridium difficile, which is an anaerobic spore-forming gram-positive bacteria. But in doing so, it probably damages other gut microbes as well. So fidaxomicin, which was produced by Astellas Pharma, is deemed to have a narrower spectrum of activity and preserve the gut microbiome better than vancomycin. But this paper showed quite definitively, although there were methodological, not concerns, but improvements which could have been made to the study, which showed broadly similar results. And actually fecal transplantation, I think now, is without doubt the most effective treatment for patients with recurrent forms of C. difficile infection. Now, with that being said, there are still many, many, many unanswered questions. And one question I wanted to ask James was, should we be screening our donors for increased risk of colorectal cancer? Because presumably, if you're transferring fecal material from one person to another, if they've got a predisposition to developing colorectal cancer, then should we be doing an FMT? But in addition to that, there are all these, and I'm not going to read them out one by one. But basically, this field has got a lot of work to do, notwithstanding the clinical evidence. Now, the proof of concept that's been achieved in C. difficile infection is, obviously, and I'm probably very biased in saying this, but it's pretty exciting. Because of the efficacy and because of the burden that this disease places on patients, it can be really pretty horrific. And we wonder if that evidence and that proof of concept can be translated into other diseases or infections associated with perturbances to gut microbiome structure and function, of which there are many, and you'll have heard lots about it already this morning. Now, we look at this and we go, well, how many of these are we confident that the gut microbiome is definitively linked to the pathogenesis and the severity of the disease? How sure actually are we that it's playing a causal role? And I would go for some, like C. difficile, I think we're pretty sure, but for others, it's just a correlation, and the most commonly observed phenomenon is reduction in diversity. So I'm going to talk about C. difficile, inflammatory bowel disease, and irritable bowel syndrome, because I was asked to talk about intestinal applications, and there's so many, we couldn't talk really about any more. So on the left-hand side of this figure, this is a kind of pyramid where we're trying to distill our thoughts and views on the microbiome's drive, if you like, in the pathogenesis and severity of disease. And at the top, the most definitive, I think, is C. difficile infection. What's happening in C. difficile infection? In essence, too many bad bugs, not enough good ones, and probably an impaired immune response to the toxins which are being produced by the toxin-producing strains. And generally speaking, people get an infection after broad-spectrum antibiotics, wipes down colonization resistance, and then that opportunistic pathogen finds a niche and proliferates faster than the rest of the bugs in the gut. And we do know that patients with C. difficile infection do have reductions in phylogenetic diversity and richness, and you can see in this paper here, when they get recurrent forms, that's exacerbated. And that explains why patients keep getting repeated forms of the infection. And that's why I believe that it's so perfectly suited to a microbiome modulating intervention. So in light of that then, how do we think FMT works? Well, again, we're not really too sure. There's a number of competing theories. Generally speaking, in my opinion, you can distill it into direct effects on the recipient's microbiome and indirect effects on the host's physiology, essentially, which could be an immune response, it could be stimulating the production of certain things in the gut. So moving forward then, IBD and IBS, which is next down on the list. I think the MDROs are very interesting, and I might, if I've got time, to talk a little bit about what we're doing in enterobiotics to try to increase the efficacy of FMT in multi-drug-resistant organism infection. So IBS then, what's going on in IBS? What is IBS? Some gastroenterologists will sort of dismiss you and go, oh, gosh, that's the bane of my clinical life. Basically, I can't really answer that question succinctly, but it's probably a spectrum of diseases where people have symptoms relating to their digestive tract, essentially. And there's a spectrum, constipation predominant, diarrheal predominant, people in between, etc. Is there a distinct microbiome profile for patients with IBS? No, and given that it's probably an umbrella term for a spectrum of different diseases, that makes sense. But this is just one paper published at Bowles et al. In microbiome, and they showed that a subset of patients had a kind of definitive kind of microbiome profile, which they could correlate with symptomatology. And reductions in diversity do feature quite widely in the literature. So in light of that, there probably is rationale for microbiome modulation, and maybe one day we'll be able to segment patients with IBS-like symptoms into a particular form of IBS associated with a consistent trend, if you like, in the microbiome. So there are some signals for FMT, and there's been 5 randomized controlled trials now published to date. There are some signals, with some studies showing an improved effect, some studies showing the placebo having a better effect, this study being one of them. So the study showed that FMT robustly increased microbiome diversity. That seems to be a good thing. But actually, there was no significant benefit over the placebo whatsoever. So the capsules did better, as in the sugar capsules did better than the FMT capsules. And I can't explain those results, I'm afraid, but we can maybe discuss it in the Q&A and the panel discussion. Here's a really nice systematic review published by some colleagues based in Gemelli in Italy. I did a fellowship there a couple of years ago, and it was excellent. And just for your interest, some of you may know this, but in some of the hospitals in Rome, they have microbiome clinics. So there's a doctor who runs this clinic, and patients come in, they go, I've got a bit of this, got a bit of that, and here's my gut microbiome test, and they go, well, we'll give you a bit of this and a bit of that, and maybe we'll give you FMT. That's happening right now in a developed part of the world. And I guess people have mixed opinions on the utility of that and whether or not we have precise and robust enough tools to therapeutically modulate the microbiome, but it is happening, and that might be something that happens in this country in the future. So let's talk about IBD then. So inflammatory bowel disease used to be thought of as a genetic disease, and that's because the biggest risk factor for developing symptomatology and the disease was your parents having the disease. But then twin and adoption studies were pretty inconclusive. And then genome-wide association studies came along, and we identified over 200 genes associated with the disease. And we went, oh, sugar, it's much more difficult and complicated than we had thought. So what about the microbiome then? Does the microbiome interact then with the phenotype or the genotype in these patients? And the answer is quite possibly. So as with some of the other diseases, consistent reductions in diversity we see in both Crohn's and ulcerative colitis, the two main subtypes, if you like, of inflammatory bowel disease. And there is a middler version or indeterminate colitis, but there hasn't been any research into that as far as I'm aware. And there are distinct, reasonably appreciably frequency occurring modulating and aberrancies in the gut microbiome of patients with Crohn's disease and ulcerative colitis. And generally speaking, they are reductions in the butyrate producing bacteria and increases in the mucin degrading bacteria. So for example, in Crohn's disease, there appears to be decreases in Faecalibacterium pausaceae and increases in certain kinds of E. coli, like adhesive invasive E. coli. So there's rationale for microbiome modulation then in inflammatory bowel disease based on that. So here's 4 randomized controlled trials investigating FMT and ulcerative colitis. There clearly is a signal. And in 3 of these studies, all of these here, there was a statistical significant benefit over the placebo. Now, the placebo choice varied between the studies. So in this study, they gave autologous FMT. And is that really a placebo? I'm not sure. We can discuss it at the panel. And unfortunately, the methodological differences between each study mean that even though there's a signal, which is reasonably robustly reproduced, it's impossible to translate those results elegantly into clinical practice. So as it stands right now, FMT doesn't feature on the care pathway for ulcerative colitis in the NHS. There is a study taking place in the UK right now called Stop Colitis, which is seeking to answer some of the unanswered questions which I presented earlier in this paper. Now, given that in inflammatory bowel disease there appear to be specific changes at the species level or strain level, does that mean that enrichment of fecal material from donors could potentiate FMT's efficacy in these diseases? So this is a graph, if you like, taken from the Moriarty paper in 2015. And you can see quite clearly that one donor, donor B, had a bigger effect size on the patient population than the rest of the donors. which is pretty interesting. And I'm sure the group are doing all sorts of analysis on those stool samples to try and figure out what the magic or active ingredient actually is. It could be because of how the study was designed as well. I'm not too sure. But we published it in AP&T about a year ago now. And what we did was we went through all the randomized controlled trials and we looked at all the appendixes and supplementary data, and we tried to decipher and pick out were there any bugs, any species, any phyla consistently associated with remission. And actually there did seem to be, broadly speaking, a trend in the Clostridia clusters, and at a sort of more granular level, many of these Clostridia clusters are butyrate producers. So that could potentially be an explanation for mechanism of action. And interestingly, actually, in animal models, these Clostridia clusters induce regulatory T cells, which presumably create an anti-inflammatory effect in the gut. Now that really is quite a tantalizing proposition, because, I mean, I think all of us would agree with me if I said that we want to one day, potentially very soon, move away from needing to transfer stool from one person into another. And we will have a more aesthetically acceptable, standardized methodology to drug the gut microbiome and produce reproducible effects in specific patient populations. Now, we're probably a long way away because it's so complicated, but this is pretty interesting because it may allow us to use fecal transplantation as a tool to reverse engineer what's happening in the clinical trials where we see a positive effect. And what you would do, in theory, and it is happening more and more frequently now, is combine these interventional clinical studies with deep dive full shotgun metagenomic sequencing of the donors and of the patients pre and post fecal transplant, with a view to identifying specific strains which are consistently associated with the phenotype of interest in that disease population. And it's something we're working on at Enterobiotics. Now, with all of that being said, I think it's fair to say that there has been a big explosion in academic and clinical interest, and also patient interest in fecal transplantation. It did extend this recently to 2018, 2019, and it is continuing to go up. So those results, especially in C. difficile infection, have prompted the patient population to take matters into their own hands, because it's actually quite difficult for a doctor to perform fecal transplantation. They've got to find a donor. They've got to screen the donor for infectious diseases. Then they've got to process the samples. And when I got started out with my venture, I remember coming across the NICE guidance, coming across the New England Journal of Medicine paper and going, whoa, that's incredible. And then came across a paper which suggested that it might help people lose weight. And I was very into health and fitness at the time. I thought, right, this is it. You know, I'm going to create the next weight loss drug, and that's it, set. Obviously, it's more complicated than that, and we went down a slightly different route. But nevertheless, the difficulties that doctors face mean that patients are doing it themselves. And we published a paper in Frontline Gastroenterology a couple of years ago that showed that 10% of UK gastroenterologists, paediatric and adult, were aware of a patient that had undertaken FMT on their own. And I'm not sure any of you are going to go home and Google this, but you can at your leisure. And you'll see that there's a large body of patients who are undertaking it themselves, both to treat C. difficile, but also all sorts of other things as well. And that's quite worrying. Now, how to safely, of course, I'm going to give my company a plug. Hope I'm allowed to do that. If not, then very sorry. But I'm here now and the slide's up, so we're going to have to roll with it. So basically, after identifying those challenges, I decided that I wanted to do something about that and wanted to create an MHRA licensed facility whereby we would do all of the work that doctors currently are doing and create an off-the-shelf, ready-to-use product. And actually, in the last 2 years, we've done that. So our facility holds 2 licenses from the Medicines and Healthcare Products Regulatory Agency. We can release products for use on a named patient basis and for use in clinical trials. We have 2 formulations at the moment available for patient use. We've distributed to Scotland, England, Wales, Northern Ireland, and Ireland, so we've done the full circuit. Basically, what we're doing with that platform is 3 things. One of which is focused on creating a lyophilized, orally administered powdered formulation of the fecal microbiota that patients can swallow, rather than having to get a tube up the backside or a tube that goes up the nose and into the small intestine. I think we all agree that although you are, in effect, swallowing fecal material, which I know I can see on the faces, that's a more tantalizing prospect than getting a tube through the top or through the back passage. We are combining the fecal material with precision antimicrobials, and the idea is to create a dual effect, if you like, in the patient population, where the precision antimicrobials will go in, clear out the pathogens, and then the good bugs will go in and repopulate and reinstate colonization resistance. and we've got some funding from the Medical Research Council to take that into preclinical development. And the final part of the company is focused on reverse engineering the fecal transplants, but with a particular focus on the small molecules that they're producing in their repertoire of activity, rather than just the bacteria themselves and trying to culture them up and create a therapeutic. So these are the products which are being used in the NHS here. And this is the capsule which will be released into a number of clinical trials later this year. And just to say, we spoke a little bit about IBS. I'm really excited about one of the trials we are doing with IBS, where we're trying to, for the first time, stratify patients who are producing too much of a certain kind of gas in their gut to receive FMT, with a view to the bugs in the FMT outcompeting the gas producers and reducing symptomatology. And as a non-invasive biomarker that we're able to use with one of our partners and collaborators to enable that clinical trial. So I don't have a conclusion slide, I'm afraid, but generally speaking, everyone just takes photographs, so maybe we can kind of hammer it home here and it will stick in your mind before lunch. So basically, FMT, fecal microbiota transplantation, is an established and approved treatment in recurrent C. difficile infection. And we are doing it now in the UK, and it's happening all around the world. It is an experimental drug, because it is a medicinal product, in inflammatory bowel disease, IBS, and probably all the other indications you saw in that pyramid, apart from the drug-resistant organisms. But there's over 150 or 200 clinical trials going on around the world which seek to basically find the next disease indication. There are unanswered questions, and there are risks associated with FMT. But I believe that it's got utility as a powerful therapeutic and as a research tool when coupled with the correct kind of microbiome sequencing and profiling. And finally, things like off-the-shelf capsules will basically improve the design of placebo-controlled clinical trials and expand access for patients. Thanks very much.